Hi, everyone. Welcome back. This week, we are going to finish off our discussion on cells, specifically looking at two main concepts. Last week, we had looked at the overall structure of the cell and then described the general functions of all the different parts. All right, so we started with the plasma membrane, looking at how it separates the external from the internal environment of the cell, so the extracellular environment and the intracellular environment. We then looked at that intracellular environment and learned that there was cytoplasm, that jelly-like substance that suspends everything within the cell. And then looked at the different membrane-bound organelles that each have their own role to play. All right, so like a machine with lots of little parts. What we mentioned briefly, but now we're going to take a look at today, are these different proteins that make up what's known as the cytoskeleton. All right, so within the intracellular space, it's not just cytoplasm and everything floating in that. There's actually different proteins that are there to provide structural support, support the shape of the cell, be allowed to help the cell move, as well as to be able to transport things within the cell. You know, we talked about, for example, vesicles coming off of the rough endoplasmic reticulum. Well, there are specific proteins that are involved in the process of actually transporting those vesicles. They're not just, floating through outer space, so to speak. Okay, so we're going to take a look at those main structural features of the cytoskeleton today. And then we're going to jump outside of the cell and we're going to look at the extracellular proteins that make up a lot of that space. Now, we've already looked at connective tissue, right? We looked at very specific types. And we know that there are three main protein fibers that you find within the extracellular space, right? So this is that extracellular space. We just happen to be looking at one generic cell here. All right, so within this extracellular environment, we're going to see collagen, elastin, reticular fibers we've talked about. So we're going to talk about some of those proteins and the role that they play as well within the extracellular environment. And then finally, how the extracellular and intracellular environments are capable of essentially being connected with one another and communicating with one another. So then we'll move from there and take a look at 3.6, where we're going to then focus on how this semi-permeable phospholipid bilayer is capable of being able to allow certain substances in and out, and the ways it goes about controlling these different processes. The internal aspect of the cell is not only composed of the cytoplasm with the membrane-bound organelles, there's also that scaffolding structure that is supporting all of the structures within the intracellular environment. There's three main different types of filaments, each made up of different proteins that are all responsible for different functions of the cytoskeleton. All right, so first off, I want you to think of the cytoskeleton essentially the same as our own skeleton, right? Cyto meaning cell, skeleton, supportive structures similar to the way that our skeleton supports us. Now, the difference here, where our skeleton is made up of bone, the internal cellular skeleton is made up of proteins. Now, some of the roles of the intracellular proteins are to anchor organelles to fixed locations, right? We talked about certain ones that are going to be in fixed locations. Some, like, your free-floating ribosomes, are just going to exist within the cytoplasm. But here's an example here. We have the rough endoplasmic reticulum. That is a membrane-bound organelle that remains in a fixed location adjacent to the nucleus, right? So those proteins can be made and then move along to be further modified. We have other microfilaments that are responsible for supporting the cell shape, and then others that can help to organize ribosomes, as well as other enzymes that are needed for many different types of activities. So you need to think of the cell, and if you haven't watched the video yet, there's one to do with the inner life of the cell. Okay, I'm going to be referencing that a couple of times this lecture. Take a look at that. It really gives you perspective as to how these proteins are actually organized and dynamically assisting in the overall function of the intracellular environment. So the three main different types of protein fibers that we have in the cytoskeleton are your microtubules, microfilaments, specifically made of actin proteins, and the intermediate filaments. So you, at this point, need to know the three main functions here associated with each type of filament. So we're going to start there at the top. So the actin filament, also known as microfilaments, are made up of individual actin proteins. And if you're looking here, it looks almost like a double strand of pearls. Each of these little green spheres, that is an actin protein. And in a couple of weeks, we're going to be talking about the muscular system, and you're going to see actin is also the protein that is involved at the cellular level at making the muscle contractions work. So here as well, within the generic cell, actin filaments play that same role. Their primary function is cell movement. The microtubules, I want you to think transport. Okay, and the microtubules, they're made up of another different type of protein. Again, it's these spherical proteins that are known as tubulin. Now, the microtubules, just as the name implies, are literally small tubes. So if we were to turn this microtubule end on, you would see a hollow tube, just sort of like a straw. And remember, this is a dynamic environment. Microtubules are going to be created and then disassembled and taken apart. So these tubes are constantly being formed, and anytime something needs to be transported from one region to the other, microtubules can be put together. One example of a microtubule that we talked about last week are the spindle fibers, right? The spindle fibers are those microtubules that extend from the centrioles from either end of the cell. If you come back here, there's your centrioles. These would be, your examples of your spindle fibers at this point. So each one of these is a microtubule. And what happens is when the cell's ready to divide and the chromosomes are all lined up in the center, the centrioles, one goes to one side of the cell, the other goes to the other side of the cell. And then each of them starts to build out those microtubules, kind of like they're fishing in towards the center. And then they grab onto all of the chromosomes, line them all up, and then they're ready to then pull them apart to opposite sides. Now, there's also many different types of motor proteins as well that are capable of binding to microtubules. And these motor proteins are capable of being able to actually bind to the microtubule and walk vesicles along the length of the microtubule to transport them from one part of the cell to another. All right, that's one thing that you can also see in that video. You get a really good picture of a motor protein that's transporting a vesicle along a microtubule. And then last but not least, we have the intermediate filaments. All right, now the intermediate filaments are slightly smaller than the microtubules. They're made up of ropes of proteins. And the whole idea here is that they are providing the scaffolding for the cell. All right, they are extremely strong, and they are supporting the cell structure and are helping all of these organelles to remain in place. And one example of an intermediate filament that we covered previously was keratin, right? So let's think about our specialized cells. Where do we have keratin? In the keratinocytes. So in those squamous epithelial cells, we would have specific intermediate filaments such as keratin. So you have your microfilaments, microtubules, and intermediate filaments, and then the main functions: movement, transport, and then structural support. Now, one way that microtubules as well as microfilaments can be used is both inside and outside of the cell. One example we've already looked at is the way in which our cells are capable of moving by essentially swimming using a flagella or cilia. Okay, now, this is an example here of a bacterial cell with multiple flagella, but the spermatozoa have a single flagella, all of them made from microtubules, so those tubulin proteins. This is known as extracellular swim, right? So one way that a cell is capable of moving. So it's a protein that is attached to the plasma membrane, which then allows it to be able to then move from point A to point B based on its extracellular appendage. Right now, another phenomenon we haven't talked about yet is known as intracellular crawl. So this is an example here of an amoeba. And an amoeba is capable of rearranging its plasma membrane to form extensions that stick out. And then with inside of the amoeba, it's capable of rearranging all of the actin protein filaments that are responsible for movement And essentially, by creating these pseudopods, which are like false feet, they're able to crawl or drag themselves along in whichever way they want to go. All right, so instead of it being some sort of extracellular force that's propelling it, it's extending these pseudopods by rearranging the actin from an intracellular perspective. And that's allowing it to essentially just like spread out like a blob. If you were to just drop paint in a big blob and you would see it just spread all over, well, that's sort of what the amoebas are doing. Okay, and there's another video there as well. No sound to it, but you can see how the amoeba is capable of moving around just using these actin filaments and rearranging them. This is an example of the transport mechanism that I mentioned there with microtubules from the intracellular environment. So there are specific motor proteins that are capable of binding to the microtubule and essentially look like they are walking along the microtubule to get from point A to point B, carrying a vesicle to get to where that vesicle needs to get to. Okay, so we're going to move to the extracellular environment now. We're going to move from looking at the structures that make up the inside of the cell and look at how the cell itself is actually protected by a number of different proteins that create almost a similar environment to that of a cell wall that you would see in plants. Now, every cell is going to be producing proteins. All right, we know the ribosomes are the protein factories. What we haven't done, though, is really put together the pieces of, okay, well, what types of proteins or where are these proteins headed? So as we know now, some of them are going to be meant to stay in the cell, right? Because there's lots of different intracellular proteins like the microtubules, so the tubulin, the intermediate filaments, and then the actin proteins. All of that needs to be produced so that the cell can continue to thrive and survive. But there's going to be other proteins that their destination is either going to be somewhere outside of the cell. And in those cases, those proteins then will be transported. Some of them will be destined for the cell membrane itself, right? We know we have structural proteins that exist within the cell membrane. So we're going to talk about those. We're going to look at those that are destined for the extracellular environment and see how all of these pieces connect with one another. So as you know, within the extracellular environment, whether it's within connective tissue, right, in the extracellular matrix, or just any other tissue type outside of the cell, we have an abundance of collagen and elastin proteins. All right, and so as we know, collagen is there to provide strength to many tissues to be able to resist, any kind of tension in different directions. And then elastin is there to provide elasticity, right? To allow the tissue to still be flexible and be able to retain its original shape. So you see different amounts of these different fibers depending on the tissue you're looking at. We know that in our tendons, for example, we have an abundance of collagen, right? In that dense regular tissue. We know in our ligaments as well, we have the same. In our fascia or in the deeper layer of the dermis, right? In that reticular layer, we have dense irregular connective tissue. So we're going to have collagen fibers there. We're also going to have elastin fibers, right? In the lower layer of the dermis to allow for that flexibility. We also see elastin in abundance within the connective tissue of our bladder because there's going to be a lot of stretch and recoil going on. Within the lungs as well, in the blood vessels, we also have elastic tissue that exists to allow the arteries to be able to vasoconstrict, so tighten up, and then also vasodilate when they're letting more blood flow through. Now, these elastin and collagen fibers then are able to be connected to the cells via specific proteins known as fibronectin, which you can see right here. Fibronectin is a protein that is responsible for connecting the cell to the extracellular environment. Right? And you can see here the fibronectin connects to both the collagen and the elastin fibers. But then fibronectin as well is then bound directly to these structural proteins embedded in the cell membrane, and those are known as integrins. Now you can see here then on the internal aspect of the cell, the integrin proteins are then connected to the actin filaments. Remember, actin filaments are responsible for movement. Now these integrins extend directly through the cell membrane. They're embedded within the cell membrane, and they connect the intracellular to the extracellular environment. And this allows the extracellular environment then to have an influence on what's going on inside the cell and how that cell is going to behave. And we're going to finish off this section taking a look now at how the cells then are going to be connected to one another in the tissue that they exist in. Okay, because remember we looked at all those epithelial tissues, and I mentioned to you there's all sorts of specific proteins that are holding those cells together. There's also cell junctions that are allowing for communication to happen between the cells. So really, the three of these different cell junctions all play their unique role in allowing the cells to form the functional tissue. So we're going to start over here looking at the anchoring junctions. Now, the anchoring junctions, also known as desmosomes, right? They consist of a specific protein that you can see here between the cell, known as cadherin proteins. And those cadherin proteins, which are located in the extracellular space, they connect directly to the intermediate filaments, which are the structural proteins inside of the cell. All right, so these desmosomes are providing that structural support and adhering the tissue together by connecting each cell to the structural framework that makes up its own cytoskeleton. So we find desmosomes in our stratified squamous epithelial tissue. That shouldn't be a surprise, right? That tissue undergoes so much stress, and so we need to make sure that those cells are well anchored to one another. We also have desmosomes in cardiac muscle tissue. And we're going to get to look at cardiac muscle tissue in more detail, but I talked about in the second week there with tissues that cardiac muscle tissue has a unique structure known as the intercalated discs. Now, intercalated discs are unique structures that are made up of a combination of desmosomes as well as gap junctions. All right, and we'll revisit this concept when we get to that point a little bit later. So the next junction we're going to look at are tight junctions, and they are made up of occludins proteins. Right now, the key here, tight junction, you're not wanting anything to be able to simply pass through that tissue. They bring cells so close together to minimize the extracellular fluid in that area. Now, why would you want to minimize or essentially put a barrier here? You want to do that because you want to prevent molecules that could potentially simply pass through and move between cells. And one of the places where you find tight junctions are within the nervous tissue of the brain, right? Because we don't simply want to have molecules simply being able to just pass through and being able to affect our nervous tissue. Now, the last type of cell junction that we have are known as gap junctions. And gap junctions are set up differently, right? Whereas the occludins and the cadherin proteins are really kind of anchoring to one another. The connexin proteins found in gap junctions form a tube, essentially a connection. So the intracellular environment of this cell is directly connected to the intracellular environment of this cell. And so this is a way that you can, if you have a certain level of, say, sodium in this cell that's higher than this cell, well, it's able to then rush through into this next cell. What we're going to learn as we get into the nervous system is that the passage of these ions across cell membranes is actually what causes the nerve impulses to occur. When you have these gap junctions occurring within the cardiac muscle cells, it allows for the electrical signals to be able to spread very rapidly from cell to cell to cell to cell. So that way it's not just relying on the neurons stimulating the cells, it's that each cell can communicate with other cells within that tissue. Okay, so the three cell junctions, the desmosomes, also known as anchoring junctions, which have cadherin proteins, okay, holding the cells together and anchoring them to the structural intermediate filaments that make up their cytoskeleton. We then have the occludins proteins that form the tight junctions. We find these in the brain. And then the connexins proteins are doing what they say, they're connecting, right? So the connexins proteins are forming those channels between adjacent cells to allow for that communication between them to take place. So now we're going to finish off our discussion on cells by looking at how their semi-permeable membrane is capable of regulating what passes in and what passes out. Now, to do this, we're going to set the stage by first learning some of the basic principles of movement of substances. All right, so we're going to look at the difference at the beginning here between diffusion and osmosis. So first off, diffusion, I want you to think simply movement, right? And movement of what? Movement of substances. So diffusion occurs when molecules move from an area, and this is key, from high concentration to an area of low concentration. Diffusion is a passive process, which means it does not require ATP. One of the underlying principles of biochemistry is that substances will always move from areas of higher concentration to lower concentration. All right, and as they do that, think of them like a fish that's just swimming downstream. Is it having to do much work really to get down there? No, it's the easy route, right? So with diffusion, you don't require too much energy. You could literally just throw a stick in the river and it's going to end up going with the flow. We're going to see the contrast where we look at active transport processes that go in the opposite direction, moving against their concentration gradient. Okay, but first we're looking at simply passive processes. So first, diffusion is the movement of substances or molecules. All right, so one example here that you can see is oxygen. So when we breathe oxygen in, it gets into the deepest parts of our lungs. And then because the oxygen in our blood is much lower around the lungs, oxygen can simply pass through and move right into the bloodstream where it's then sent throughout the body. All right, so that's one example of diffusion, passive transport from high concentration to low concentration. Now, osmosis is also a passive transport mechanism. But with osmosis, we're looking at the movement of water. And in this case, water is always going to move from an area of higher water concentration to an area of lower water concentration. So diffusion, you're looking at the molecules themselves moving. With osmosis, we're looking at where is water going to move. And we're going to take a look at some different examples of different solutions and circumstances under which water is going to want to either move into our cells or out of our cells. Now, a quick little reminder in case I haven't already reminded you enough. Cell membranes are composed of these little guys. Those are the phospholipids. And remember, phospholipids are amphipathic. What does that mean? Amphi, both. So they have both hydrophobic and hydrophilic regions. The tails, which are made out of fatty acids, are hydrophobic, and the polar heads are made up of a phosphate group. All right, and that's what gives them their amphipathic nature. So they have a hydrophobic region in the tails, and the head is hydrophilic. They arrange themselves to form a hydrophobic interior to that membrane. Only certain molecules are capable of simply crossing the membrane, moving from the extracellular environment to the intracellular environment. And one of the exceptions is large molecules and charged molecules have to cross the membrane using specific proteins. They can't just simply pass through. All right, so we're going to look at the differences now of the different substances that can simply move through using diffusion, whereas then later we're going to take a look at those that require the use of some sort of mechanism to be brought into the cell. So as we mentioned, certain substances can diffuse across the plasma membrane, but they have to be small and they can't be charged. All right, so a couple examples here, molecules, which we'll go through a couple of them there, and water. All right, so remember, do these all fit the criteria? Yes, they do, right? They are all relatively small molecules, CO2, 3 atoms, oxygen, 2 atoms, nitrogen gas, 2 atoms. Glycerol is a little bit bigger, but it is not charged, so it's capable of moving through. And then water. Water is polar though, so it kind of breaks the rule here, right? But it's the exception because it is such a small molecule, it is capable of passing through. But the way that water typically will move through the membrane is through specific aquaporin channels. All right, and when we get to the urinary system, we're going to talk about how the antidiuretic hormone that's released from our pituitary gland is responsible for allowing us to be able to retain more water by having these aquaporin channels made in the kidney to allow us to reabsorb more water and have less go out in our urine. So these aquaporin channels are specific protein channels that allow for water to simply pass through when they're open. So as I mentioned, ions, which are your charged atoms, right? Your charged elements, your sodium ions, chlorine ions, potassium ions, you name it. If it has a charge, it cannot pass through. Polar molecules, except for water, cannot pass through the hydrophobic environment, and larger molecules can't as well. They require specific membrane proteins, whether they be channels or whether they be co-transporters or pumps that are going to bring them into or out of the cell. Now, as we looked at before, diffusion, remember, being just simply the movement of molecules into or out of the cell depending on the concentration gradient. So in this case here, what you're seeing is the intracellular fluid is lower in oxygen and the extracellular fluid is much higher in oxygen. So you know oxygen is going to diffuse across the membrane to the intracellular space. Here's an example just using some food coloring. Add that to water. These molecules are going to continue to move from an area of high concentration to low concentration until finally they are equally dispersed throughout the solution. Now, there's still going to be movement, right? Everything doesn't just stop moving when things are balanced, but there's not going to be any net flow of direction, okay, once all of the molecules have been distributed. Now, as we mentioned before, remember osmosis now, we're looking at the movement of water, and it's going to move down its concentration gradient as well by moving into or out of a cell. Now, this all depends on the solution of the extracellular fluid compared to your intracellular fluid of your cell. All right, so the greater amount of solutes that are dissolved into a solution, the less amount of water molecules that are free to move. So let's talk about this. If we have, say, a 3% solution of sodium chloride, and then we have another solution of 5% sodium chloride, where is the water content higher? The water content will be higher in the 3% solution because 3% is sodium chloride, 97% is water. In the 5% example, 5% would be sodium chloride and 95% would be water. Okay, so just some definitions here, which you're going to cover in chemistry. A solute is the substance that has been dissolved. So in this case here, we're looking at a solution with 3% salt. So in this case, the salt would be the solute. And the solvent would be the medium in which you have dissolved that solute. So in this case, it would be the extracellular fluid, okay, or water or anything that you're dissolving that substance in. The concentration of all molecules that have been dissolved in that solution is called the osmotic concentration. Now, osmotic concentrations of different solutions then can be compared to each other. So here we have the intracellular space or compartment, and that's going to have its own osmotic concentration. And then you're going to have the extracellular space or fluid. And that extracellular fluid is going to have its own osmotic concentration. So if the inside of the cell has a higher osmotic concentration than the outside of the cell, then water is going to want to move to an area where it is less concentrated. All right, so based on the concentration of solutes in solution compared to the intracellular space, we have a number of different scenarios that can play out. All right, so a hypertonic solution is a solution with a higher concentration compared to the intracellular concentration. All right, so here's an example. Your cellular concentration of sodium chloride is about 0.9%. All right, so here you can see that. You would have 0.9% inside the cell, and in this case, the solution itself outside is also 0.9. So that's an example of an isotonic solution, where the solution is equal to the osmotic concentration inside the intracellular space. Now with a hypertonic solution, it's higher in the solution than it is in the intracellular compartment or space. So in that case, if it is more salty out here than it is in here, water is going to want to move to the more salty environment, right? Because it's going to move from the area of higher water to the area of lower water. Now here, if we have a hypotonic solution where the solution itself is a lower concentration compared to the intracellular environment, you're going to have the opposite effect. Okay, so so here we're looking at pure water. So pure water would have no salt in it. So you have 100% water outside of the cell, and inside of the cell, you have 99.1% water. So water then in that case is going to want to move from outside to inside of the cell. Well, what's a problem there, do you think? As soon as you have that net inward movement of water into the cell, there is a limit there, right? Cells cannot continually just getting bigger and bigger and bigger. We talked about that. Eventually, they can burst. So this is why when you're running a marathon, they don't just give you straight water to keep replenishing your fluids, because as you sweat out all of your sweat, you're going to be losing all sorts of solutes, all sorts of salts, right? And you need to replenish your body with those. They're known as electrolytes. Right? That's what it is. Dissolved salts are electrolytes. You need to replenish your body with those. So they don't give you pure water when you're running a marathon. They give you some sort of Gatorade or electrolyte substance or drink that's going to help to replenish those levels, so we don't end up with a situation where we have our cells completely swelling and ultimately reaching a point of water intoxication. So here are 3 examples here. We have hypotonic solution, which is lower than the intracellular space. We have an isotonic, so iso meaning same. Isotonic is the same concentration. So in this example here, it's balanced, right? There's going to be no net movement of water. And this is what you want to do. Say if you're giving a patient a saline drip, right? The reason that we give them that saline drip is to maintain their extracellular fluids with what the intracellular fluid levels would be. All right, and then here's another example here where you have a hypertonic solution. So let's just say we go and eat a whole bag of salty chips, right? We increase the amount of salt in our extracellular fluids. Well, then the water is going to move out of the cells. Right? So that's how you can become essentially dehydrated. And that's why when you've eaten a whole bag of salty chips, I'm not saying you've eaten a whole bag, but you're also going to want, or you're going to be thirsty, right? You're going to want to have some sort of beverage to help balance that out. So as I mentioned before, based on what that solution is, if your extracellular environment is hypotonic, that's going to cause the water to then move into the cells. So eventually cells can swell and ultimately burst. If you're looking at the opposite effect here from a hypertonic solution, if you eat that bag of salty chips, all right, and you haven't drinking any water, well, you end up with shriveled cells. If you're giving a patient saline drip or you're taking in, electrolyte beverage that's balancing everything out, then you have no change. So those were 2 examples of looking at passive processes. We looked at diffusion and we looked at osmosis, right? With both of those, they're passive, which means you are looking at movement from high concentration to low concentration. Now, this is another example of a passive transport mechanism, but requires the use of some sort of membrane protein to assist in transporting that substance. So here we're looking at the transport of glucose from outside of the cell to the intracellular environment. And why do we need glucose inside the cell? That's so we can metabolize it through glycolysis, Krebs cycle, and make ATP. All right, but to get glucose into the cell, it's still moving down its concentration gradient. So it binds to the membrane protein. The membrane protein changes shape and brings it into the cell. All right, so that's facilitated diffusion. So diffusion, simply the molecule is capable of just passing through, but for those substances that are moving still with their concentration gradient from high to low, but are too large, they require the use of a specific protein. So this membrane protein brings, for this example, glucose into the cell using facilitated diffusion. Now, there are a few different ways in which facilitated diffusion can take place. The first one is simple protein channels. So a protein channel is an open passageway for whatever is small enough to fit inside. And channels are specific. If you're letting a certain ion pass through, it's only that one ion that will pass through. So there are specific sodium channels, specific potassium channels. Some of them are voltage gated, which means they only open when the membrane reaches a certain membrane potential. Right? That's how the nerve impulse is created. And we'll talk about that later. For now, just understand that the protein channels are going to be specific conduits through which a specific ion is capable of moving from the extracellular to the intracellular environment. So we're looking at facilitated diffusion still. So it's again moving with its concentration gradient. And then the example we gave before with the protein carriers that are going to bring in the, for example, the glucose molecule into the cell. Now, with the carriers, there's always going to be a limit. So we do have a limit at how much glucose we are capable of bringing back in to our cells, and that's limited by the number of proteins available in that given membrane. Now, cells are capable of maintaining concentration gradients between their extracellular and their intracellular environments. And two of the ions we're going to be taking a lot more detailed look at going forward are going to be sodium ions and the potassium ions. And you can see here, the sodium ions exist in a much higher concentration outside the cell, and potassium ions exist in a much higher concentration inside of the cell. So I want to ask you, if we all of a sudden put a channel in here that opened up just for sodium, where is sodium going to move? It's higher here, lower here. Right? So right away, based on that principle, you know sodium is going to want to flow into the cell. Vice versa, if you put a potassium channel right here, potassium is going to want to move out of the cell. All right, and it's that movement of these ions across the membrane that actually causes that electrical potential to occur. Now, the way that the cells maintain this gradient, with sodium higher outside the cell and potassium higher inside the cell, is through the use of an active transport mechanism known as the sodium-potassium pump. All right, all of our cells have sodium-potassium pumps, and all of our cells maintain these gradients of higher sodium outside and higher potassium inside. And so because this is active transport, we require the use of ATP, okay? So this requires energy. Why? Because we are actively pumping sodium out into a region that's already high in concentration. And we're going to be pumping potassium into the cell, which is going to be moving it also against its gradient into a high concentration zone. All right, so because think of it as salmon swimming upstream, right? Trying to get to their spawning grounds. They are going to be having to work so hard because they are not going with the flow anymore down the stream. They are now actively going against the current. All right, so it's the same thing here with the active transport mechanisms. Sodium is pumped out of the cell, potassium is pumped into the cell. And specifically, there are 3 sodium that are going to be pumped out for every 2 potassium that are brought in. Now, to give you perspective, nearly about 1/3 of the energy that's expended by your body's cell is just given to driving these sodium-potassium pumps every day. So if you take a look and think about if you have a roughly 2,100 calorie diet, you're looking at 1/3 of that's going to be about 700 calories a day, just so all of your sodium potassium pumps are capable of working. Now, another active transport mechanism, this we're going to look in a lot more detail when we get to looking at how the mitochondria produce ATP. Again, we're looking at active transport mechanisms. So, right away, we are moving against a concentration gradient. And in this case here, we are moving protons. So protons are hydrogen ions. Okay, and eventually you're going to talk about acids and bases in chem, and you're going to know that acidic solutions are very high in protons and basic solutions very low in proton concentration. Here, what we are trying to do is to create a intermembrane space. Remember, the intermembrane is the space between the outer and the inner. Within this intermembrane space, we want to pump protons to create a concentration gradient where the protons are very high in that space. And the reason that we want this concentration gradient is so that later on in the electron transport chain, we are capable of allowing them to simply pass through a process known as chemiosmosis, where they are passed through the ATP synthase protein, brought through into the matrix. Okay, this is the matrix. So from intermembrane, brought into the matrix. And through that whole process, an ATP is created. So it's expending energy to pump the protons across. There's 3 different proton pumps that do this using the electrons that we've transported from the Krebs cycle. And now all these protons are building up in the intermembrane space. And then finally, the ATP synthase is going to allow for those protons to be able to pass along their concentration gradient, and during that process, creating an ATP through phosphorylation. Now, lastly, we're going to take a look here at coupled transport mechanisms. So one of them that we already looked at were these sodium-potassium pumps. Okay, this is an example where sodium transport is going to be coupled with potassium transport. So for every 3 sodium that are pumped out of the cell, 2 potassium are brought back into the cell. Remember, requires ATP. Now, coupled transport is also known as cotransport, and another secondary active transport mechanism that is utilized is the process by which we can bring sugar back into our cells. And this is coupled with the transport of sodium back in. So to bring sugar in, we'll also need sodium. And because, remember, sodium has been pumped out by the sodium-potassium pumps, we have a high concentration gradient of sodium. So there's going to be a force driving that sodium back into the cell, and that's what's then going to couple with the transport of your sugar. As well, amino acids can also be transported into the cell through this mechanism. So in this case here, when you have 2 molecules that are moving in the same direction, that's known as symport. But when you have molecules moving in opposite directions, such as the sodium-potassium pump, that's known as antiport. Now, cells are also capable of moving large molecules across the cell membrane, and they do this using vesicles. Okay, remember vesicles? There's transport vesicles that we've looked at already that transport proteins from the endoplasmic reticulum to the Golgi, and then finally, out of the cell or to different destinations within the cell. But there's 2 different main processes. There's endocytosis, where the cell takes in, and then there's exocytosis, where the cell is getting rid of, right? It's secreting into the extracellular environment. So with endocytosis, the cell is engulfing some sort of substance from outside of the cell, and it's bringing it inside the cell to use it. And with exocytosis, which you can see here in the C diagram, the cell is getting rid of, it's secreting something. So for example, any of those proteins that are destined for the extracellular space that have been created inside the cell, they're going to be transported through some sort of secretory vesicle, which is then going to fuse with the plasma membrane, and its contents will be released. Now with endocytosis, there's actually 2 different types of endocytosis, and that's based on what the cell is bringing in. So if the cell is going to be bringing in solid matter, then the process by which it brings it into the cell is known as phagocytosis, right? So we talked about that in terms of immune cell functions, right? Your Langerhans cells are going around engulfing foreign pathogens. Really, any solid matter is going to be brought into a cell using phagocytosis, right? So phage, to eat. The opposite here, to bring in liquids with, say, dissolved solutes or something into the cell, that's a process known as penocytosis. All right, so phage meaning to eat, peno meaning to drink. And that brings us to the end of our lecture for this week. So we have now wrapped up cells, and we are going to be moving into looking at how our cells are capable of taking that glucose that we've talked about and creating ATP. All right, so that will be our next week's material, and that will actually be the last material before our midterm exam. All right, so if you have any questions in the meantime, please let me know. Otherwise, I look forward to seeing you all in tutorial. Take care.